afternoon, everyone. Thank you so much for attending this session. I'm really excited to present to you my intent to submit for the Master's by Research program. Like Dr. Willingham said, the title is Detection and Molecular Characterization of Different Viruses in Various Host Species from the Caribbean Region. Okay. So I will have 3 chapters to present today. The first chapter, the title of the first chapter is actually from the journal article we published in Transboundary and Emerging Diseases this year in April 2020. And the title is High Rates of Detection and Complete Genomic Analysis of Porcine Circovirus 2 in the Lesser Antilles Island of St. Kitts, Identification of PCV2B, PCV2B recombinants. So to give you a brief introduction about this virus, it is a part of the family Circoviridae, and it is considered as one of the most economically important viruses for the pork industry worldwide. It is considered as the major etiological agent of porcine circovirus disease and porcine circovirus associated diseases that collectively represent many clinical manifestations of PCV2 infections. These diseases include post-weaning multisystemic wasting syndrome, in which pigs can present with anemia, weight loss, diarrhea, jaundice, lymph node enlargement. And porcine dermatitis and nephropathy syndrome can be seen. Porcine respiratory disease complex can be seen with reproductive failure, enteritis, and congenital tremors as well. Now I want to delve into the genomic organization of this virus. PCV2 is a small, single-stranded, non-enveloped circular virus that contains an ambisense DNA genome of 1,766 nucleotides. And this is depicted here over on the right. And the open reading frame 1 is encoding for the replicase protein, while the open reading frame 2 encodes for the capsid. And it is with this capsid that PCV2 strains can be classified into at least 8 genotypes, PCV2A through PCV2H. Among these 8 genotypes, PCV2A, PCV2B, and PCV2D are currently prevalent worldwide. In about 2004 to 2005, PCV2B emerged and replaced PCV2A as a predominant strain. And then in about 2012, PCV2D emerged. So pig farming contributes significantly to the regional livestock economy of the Lesser Antilles, and there is an increasing demand for local pork in most of these islands. And although PCV2 has been reported worldwide, there are no reports on the detection or molecular characterization of this very important porcine virus. To date, with a PubMed search, one study presents from Trinidad that reported antibodies against PCV2 in pig farms. So that was the objective of this chapter, was to detect and completely analyze the genome of PCV2 strains circulating here in the pigs on St. Kitts. So sampling of the feces actually occurred before my time. This was in August 2015, and 63 direct fecal samples were collected from piglets and adults at 21 pig farms, and this is depicted by the map to the right. Some of the piglets were noted to have poor growth and reduced weight gain, and it was also noted that 4 samples tested positive for rotavirus A. So this will become a very familiar schematic throughout my presentation. Since I'm presenting it to you for the first time here, I will go into a little bit more detail of what we did. So as I said, those fecal samples were collected before my time, so we pulled those out of archive samples from the 8 degrees Celsius freezer, and we began by fecally homogenizing the samples in order to then extract viral DNA. From there, we wanted to screen for PCV samples. So we used qPCR as well as PCR to confirm those results. Here you can see what an example would have been like using PCR and then using gel electrophoresis to view our amplicons. From there, we gel purified our PCR products and sent for sequencing to analyze the data. And now I will move into the specific primers that were used for both the qPCR and then the conventional PCR. So here we see the primer and Tachman probe that were designed based on nucleotide sequences of open reading frame 2. And with qPCR, we detected 27 positive PCV2 samples from 63 fecal diaric samples. And now, as I stated, we then use conventional PCR to confirm those results from the qPCR. And in this part of the study, we designed primers that were based off of a multiple alignment from conserved regions of several PCV strains. And you can see that the reverse primer started at 372 nucleotides, whereas the forward primer started at 1,107 nucleotides, creating a 1,056 base pair amplicons, as you can see we saw and is depicted on this gel electrophoresis. So from here, we then wanted to move forward and look at the complete genome of PCV2. So for the fecal samples, based on available sample volumes, we chose 9 PCV2 positive fecal samples representing the different farms across St. Kitts for whole genome sequencing. Now at this time, we thought it would be nice to add in an additional part of the study to enhance the study by looking at kidney samples and to see if PCV could be detected from kidney samples. So again, before my time here on the project, between July 2012 and March 2013, kidneys with gross lesions where there was foci of pale and red discoloration throughout the cortical surface were collected from 61 apparently healthy pigs that were slaughtered at the St. Kitts abattoir. And again, by qPCR, PCV2 DNA was detected in 20 out of 61 renal samples. So similarly to the fecal samples, based on available renal sample volumes, 9 PCV2 positive kidneys were chosen to be submitted for whole genome sequencing. Okay, so moving on to the process in which we completed the whole genome. Ultimately, this was completed by 3 separate overlapping nested PCRs. To start, we used primers based off of the conserved regions following multiple alignment of the complete genomes of several reference PCV2 strains, representing different PCV2 genotypes. These primers were designed in such a way that there was overlap between the primers, stitching together this circular DNA virus into 3 parts. So the first PCR primers are listed here in each of these boxes. And when we viewed the amplicons from these first PCR primer sets, we noted low molecular weight bands that we suspected would interfere with the sequencing results, and lo and behold, they did. So we then designed and created nested PCR primers that helped us essentially clear up those sequencing results. And as you can see from these gel electrophoresis snaps, they're nice and clear and bright bands. And ultimately, a total of 18 PCV2 strains were sequenced for the complete genome in this way. 9 from the fecal samples and 9 from the samples. Okay, so genotyping of PCV2 is based on the neighbor joining method for creating the phylogram, as well as looking at the open reading frame 2. However, since the neighbor joining method is not as robust as the maximum likelihood method. In this study, we did phylogenetic analysis. So since the neighbor joining method isn't as robust as maximum likelihood method, we decided to create phylograms based off of both of these methods. So to start, this phylogram is looking at the ORF2 of our 18 St. Kitts porcine circovirus 2 strains. And you can see that 16 of our strains clustered with the PCV2B bracket. And additionally, outside of the PCV2B bracket, identified as PCV2B, PCV2D recombinants, we have 2 strains labeled F6 and F60. Now, this was looking at the ORF2 region. When we look at the complete genome, we now have an additional strain that has identified to be PCV2B, PCV2D recombinants, and that is MJ8 strain. Again, we also use the neighbor joining method to create a phylogenetic analysis, and the clustering patterns are similar to that of the maximum likelihood method. I'll just show it here for completeness, and this is showing the ORF2 region, and then this is showing the complete genomes. Again, similar clustering patterns as the maximum likelihood method. So this is further validating the results from the phylogenetic analysis. We see here the nucleotide sequence identities of the St. Kitts PCV2 strains, looking at specifically just the ORF2 and then specifically at the complete genome. Here, I would like to point out the conserved motifs that we saw at the putative capsid protein at the amino acid position 86 to 91. And here we see all of our strains, and this conserved motif is seen in specifically PCV2B strains that distinguishes it from PCV2A and PCV2D. And now I would like to delve a little more into the recombination analysis. So we used this program called RDP, which is Recombination Detection Program. And the theory states that if the p-value is less than 0.01, and there are 2 or more methods that have this p-value of 0.01, then we can confirm and prove that a recombination event occurred. So this is specifically looking at F6 strain, and we see here, hopefully you all can see my arrow, that the recombination event occurred in the ORF2 region around position 883. Now this was a familiar and similar story for strain F60, where we also saw the recombination event occurring in the ORF2 region. Now, in contrast, MJ8 did not have its recombination event in ORF2, but had its recombination event outside ORF2, as depicted here. And this further validates the recombination events that has been shown through the phylograms as well as the RDP program. This is a simplot recombination analysis, again showing the recombination events in all 3 of those St. Kitts strains. Now I would like, sorry, this got in the way. Now I would like to show you the 3' terminal sequence of ORF2 of the PCV2D strains and the St. Kitts recombinant PCV2B, PCV2D strains, F6 and F60. And they contained 3 additional nucleotides, shown here in green, that resulted in an extra amino acid, lysine, on the tail part of the putative capsid protein. Now, this additional extra amino acid is believed to help PCV2D strains bind more efficiently to the host cell, making PCV2D more virulent than PCV2B. Sorry, I was pushing buttons. Okay, the capsid sequences of 7 of our strains, PCV strains, exhibited a mutation, and this was in the capsid sequence that, to our knowledge, has not been reported before in PCV2. And this is shown here with the blue arrow and the green highlight, and this is showing a mutation from valine to leucine, 2 amino acids. Now they are both hydrophobic, so this switch may less likely induce a change. But since it hasn't been reported before, we still note it here because it's an interesting finding. So, okay. Brings me to my conclusions for this section, and to our knowledge, the first report on the detection and genotyping of PCV2 strains from the Lesser Antilles. The high rates of detection and widespread PCV2 in the present study is quite intriguing, as St. Kitts is an isolated island. Most of the PCV2 positive fecal samples were from piglets where 15 animals exhibited stunted growth or weight loss. And although the PCV2 positive kidneys were from slaughter-age, clinically healthy pigs, microscopically, various degrees of inflammation, mild, moderate, or severe, were observed in the 18 kidneys. PCV2B was the predominant genotype on St. Kitts, and it was identified as the sole PCV2 genotype in Cuba as well, the only other Caribbean island around us, which is in the Greater Antilles, to report PCV2 so far. Therefore, PCV2B may be a major genotype in the Caribbean. And although we didn't detect PCV2D strains specifically, the identification of those 3 PCV2B, PCV2D recombinants indicate the presence of PCV2D on St. Kitts. And these findings emphasize the importance of future studies on the surveillance and genotyping of PCV2 in other Caribbean islands of the region. And these are my references for this chapter. And now I would like to move on to chapter 2, which is also the same title from an article that we published in Viruses this year. And the title is Detection and Molecular Characterization of Picovirinaviruses in the Mongoose, Identification of a Novel PBV Using an Alternative Genetic Code. To introduce you to the virus, Picovirinaviruses are the sole member of the family Picovirinaviridae. It's a wide variety of mammalian and avian species, reptiles, and sewage samples. They are believed to be opportunistic enteric pathogens detected widely in fecal samples and intestinal contents. But it is noted that they have been reported in the respiratory tract of humans and pigs in the plasma of sick horse. And then to get into the organization, the genomic organization of the virus, they possess a bisegmented double-stranded RNA genome with the gene segment 1 encoding the viral capsid protein and a fugitive polypeptide of unknown function. Gene segment 2 codes for the viral RNA-dependent RNA polymerase, or RDRP. And it is based on the variations in gene segment 2 that PBVs are classified into genogroup 1 or genogroup 2 strains. Most PBV strains belong to genogroup 1. Although PBVs have been reported from a wide variety of host species, so far to date, there are no reports on the detection and genetic makeup or diversity of PBVs in mongoose. So that was the goal of our study, was to detect PBVs in the small Indian mongoose on the island of St. Kitts. And we also wanted to molecular characterize gene segment 2, or the putative RNA-dependent RNA polymerase of PBVs from mongoose. So this is a map depicting the trapping areas in which mongoose samples were taken from. And this was actually a part of a microbial ecology study done from Dr. Becker. And this map was actually created by her as well. So in this map, the blue dots represent the wild trapping areas where 25 samples were taken from. And then the red dots represent the urban trapping areas where 57 samples were taken from. Again, here's my schematic that you're growing accustomed to. The only thing that has changed here after we received the samples from the 8 degrees Celsius freezer, we extracted viral RNA, and then we used RT-PCR to screen as well as complete the genome, or complete the RDRP for the PBV. And I will get into those primers that we used here in a few minutes. So again, I briefly mentioned that the fecal samples came from a study during 2017 in which we received 82 fecal samples from dead mongooses on St. Kitts. And so the process of screening these fecal samples began with the use of previously published primers by Rosen et al., in which 300 base pairs are amplified if the samples are positive for genogroup 2 PBVs, and about 200 base pairs are amplified if the samples are positive for genogroup 1 PBVs. For our samples, all fecal samples were positive for genogroup 1 PBVs at a detection rate of 35.36%, and all were negative for genogroup 2 PBVs. So out of the positive samples, 19 of 57 were from the urban trapping sites, and 10 out of 25 were positive for genogroup 1 PBVs at the wild trapping sites. Okay, so at this point, we had 200 base pairs sequenced. But since we wanted to look at the complete or nearly complete portion of gene segment 2, 200 base pairs is not significant enough to draw conclusions on the genetic diversity of this virus, since the RNA-dependent RNA polymerase, or gene segment 2, is 1,700 base pairs long. So what to do? Well, through the use of different primers and their combinations, including those that I have just mentioned previously, Rosen et al., as well as the Malik et al. Primers that I will go into detail, along with a primer that we designed in this study that we labeled CON3, we were able to amplify a significant portion of gene segment 2 PBVs. So this began with a modified non-specific primer amplification method. So we ligated a non-specific primer to the 3' end of both strands of gene segment 2 of PBV strains using a T4 RNA ligase enzyme in an overnight ligation reaction at 8 degrees Celsius for 16 hours. Eventually, when the ligation reaction was completed, the ligated RNA was gel concentrated and purified. Next, we use the 1200 base pair forward and reverse primer that has been published by Malik et al. That I mentioned, and these authors designed this primer from a conserved stretch of PBV following a multiple alignment, and these primers amplify around 1200 base pairs of gene segment 2. So what we did was we used the combination of 1.2 forward primer and the complement to the ligated primer at the 3' end, and similarly a complementary ligated primer at the 5' end and the 1.2 reverse primer to amplify both the 5' and the 3' ends of gene segment 2. Now this worked really well for strain 58. You can see here that the gene segment 2 was 1,689 base pairs. And it had an open reading frame that stretched from nucleotide 47 to nucleotide 1,639. And it also retained the 5' and 3' end sequences conserved in gene segment 2 of PBVs. And this encoded a putative RP of 530 amino acids long. Since the single primer method is very complicated and time consuming, and it requires quite a bit of RNA volume to start the whole process. So to help us with the remaining strains, we instead designed a new primer, PrimerCon3, you can see here, that was based on the multiple alignment of complete gene segment 2 of our strain 58 with other PBV genogroup 1 strains. And based on this, we designed Con3. The position of Con3 is in the 3' untranslated region, which is almost near the end of the 3' end of gene segment 2. It's about 20 nucleotides upstream. So we first use the 1.2 forward and 1.2 reverse primers to amplify about 1,200 base pairs. Then we designed an internal primer with the Con3 primer to get nearly all of the 3' end, minus those 20 nucleotides or so. For the 5' end, we were still able to use the classical single primer method in combination with the internal reverse primer. With these two methods that I've shared with you, we were able to obtain the complete RDRP of 7 strains and the complete gene segment 2 of 1 strain, strain 58. So this slide is actually specifically representing one of our strain 33, to just show as an example of what we saw. And then this slide is also demonstrating a different strain in which we use the PrimerCon3 method, and that was for strain 67. So here you will see the analysis of the strains that were just characterized for the 201 base pairs, and it shows the percent identities between themselves at 65 to 100%, and that with other PVVs at 49 to 100%. And then this table shows the 8 strains that were characterized for the complete RDRP, with strain 58 being the complete gene segment 2, and the other 7 being nearly complete gene segment 2. And this table shows 48 to 99% identity between themselves, and a maximum of 51 to 85% identity with other host species. This is representing the 5' region of gene segment 2 of our 8 mongoose PBV strains in which the terminal sequence is proposed to be crucial for the initiation of the virus replication. That is shown here in yellow. And then we also see on this slide the prokaryotic motif that is the ribosomal binding site sequence that are conserved in PBVs. And the presence of this prokaryotic motif suggests that PBVs may actually infect prokaryotic cells. And upstream from so this prokaryotic motif is upstream from the initiation codon, in which you see the majority of our strains use an ATG initiation codon, whereas our strain M17A uses a GTG initiation codon, which I will delve into in the next slide. So the complete ORF of gene segment 2 of 7 mongoose PBV strains translated the putative RDRP using the standard genetic code. Now, surprisingly, the mongoose strain 17A that was detected as a mixed infection with another PBV strain, 17B, from the same fecal sample, use an alternative mitochondrial genetic code. And it could use an alternative genetic code of mold or invertebrates for translation of the RDRP instead of the standard genetic code. And this is the multiple alignment of the complete RDRP deduced amino acid sequences of the 7 mongoose PBV strains that use the standard genetic code. And you can see how they retain the 3 domains that are conserved in PBVs, which are shown in gray, highlighted gray. And the boxes are conserved polymerase motifs. Interestingly, 17A also shared the conserved domains highlighted in yellow, but it was shown to be able to use the alternative genetic code, that of mold, shown in red, or the alternative mitochondrial genetic code of invertebrates, shown in green. So to create this phylogenetic analysis in this phylogram, the alternative genetic code of mold was used, since the theory is that PBVs may be prokaryotic viruses. Now, all 7 strains that use the standard genetic code clustered together in the genogroup 1 bracket, and the mongoose 17A clustered near the genogroup 1 bracket. So our strains are the ones in the bullet points, the black bullet points. The PBVs that use alternative genetic code for translating the RDRP have been reported from invertebrates and bats and form a separate cluster from PBVs that use the standard genetic code. And those are these ones down at the bottom with the blue squares. It is intriguing that mongoose strain 17A and a strain from a bat and one strain from crustacea that uses the alternative genetic code clustered with PVs that use the standard genetic code. So you can see that our mongoose strain M17A is right here, and then the bat and the crustacea strains are right here in red, in case you can't see my cursor, which I don't think you can. This again suggests that PVVs may not be viruses of vertebrates, but rather viruses that infect fungus or prokaryotes. So this brings me to my conclusions for this chapter. To our knowledge, this is the first report on the detection of PBVs in mongoose. We reported high rates of detection at that rate of 35.36% of genogroup 1 PBVs in non-diarrheic fecal samples from the small Indian mongoose. In addition, we identified that novel PBV-like RNA-dependent RNA polymerase gene sequence that uses an alternative mitochondrial genetic code, that of mold or invertebrate, for translation. The complete or nearly complete gene segment 2 gene sequences of 7 mongoose PBV G1 strains and the novel PBV-like strain were obtained by combining a modified non-specific primer-based amplification method with conventional RT-PCRs, facilitated by the inclusion of that new primer targeting the 3' untranslated region, or CON3, as I told you. The mongoose PBV and PBV-like strains retain the various features that are conserved in gene segment 2, or the RDRPs of other PBVs. However, high genetic diversity was observed among the mongoose PBVs within and between host species. The presence of this prokaryotic ribosomal binding site in the mongoose PBV genomes and analysis of the PBV-like RDP gene sequence that uses an alternative mitochondrial genetic code, especially that of mold, for translation, validates recent speculation that PBVs may actually infect prokaryotic or fungal host cells. And these are my references for this chapter. And lastly, I want to present to you chapter 3. And I just want to preface this chapter by saying that chapter 1 and chapter 2 had a lot of data and analysis to be completed, and my master's project mostly focused on those 2 chapters. This chapter still displays some important information. It's just not as robust as chapter 1 and chapter 2. But nevertheless, the title of this chapter is Detection and Molecular Characterization of Norovirus and Cephalovirus in Children with Vomiting and Diarrhea on the Caribbean island of St. Kitts. So I'd like everybody to imagine the perfect Caribbean vacation. Doesn't this look so lovely, especially in these times during the pandemic? Imagine floating across the sea with the sun shining. Then the captain yells, "Iceberg ahead!" This isn't the Titanic, though. This is an iceberg of the norovirus. And this is actually not too dramatic. This has happened to the Royal Caribbean cruise line where the passengers aboard fell sick due to norovirus. And norovirus doesn't stop there when it strikes out. This is a publication showing norovirus in a U.S. Virgin Island resort where the residents became ill. This was based on the case definition. So, since I will be speaking about norovirus for the first 2 cases, I want to introduce you to this virus. It is considered as the leading causes of viral gastroenteritis in humans, and it is responsible for millions of illnesses and thousands of hospitalizations and deaths in the US alone. It is a common cause of non-bacterial outbreaks, diarrheal outbreaks, and it can be transmitted directly by the fecal-oral route or through exposure to aerosolized vomitus and through contaminated food, including seafood, water, and fomites. Noroviruses are a member of the family Kleymann, and they are single-stranded positive sense RNA virus. And the human noroviruses primarily belong to genogroup 1, genogroup 2, or genogroup 4. And there is the zoonotic potential of noroviruses, in which So although episodes of norovirus-like gastroenteritis based on the clinical signs have been observed in the Lesser Antilles, based on a PubMed search, there is no report on the detection and molecular characterization of noroviruses from this part of the world, including St. Kitts and Nevis. So that was the objective of this study. So this brings me to case 1. It is that of a boy that presented with nausea, repetitive vomiting, and diarrhea. And we were able to collect a sample and we labeled it RD12. And that was a diarrhea fecal sample. Here is that schematic that we are all very familiar with. I will go into the primers that we use because that is the only difference here in this part of the project. So the primers that were used to detect norovirus belong to these universal primer pair P289 and P290 that detect viruses belonging to different genera under the family Kleymann. These were used in RT-PCR assay. This primer pair amplifies a short portion of the ORF1 that encodes for the RDRP of Kleymann viruses. The amplicon size for norovirus is about 319 base pairs, which is about what we saw on gel electrophoresis. When submitting this strain for sequencing and blast analysis, RD12 was determined to be genogroup 2 norovirus strain. Now, to further genotype the norovirus RD12 strain, we used primer pair MON431 and MON433 that are used to amplify a short region of ORF1 used for genotyping. And the amplicon size is about 213 base pairs, which is what we saw on our gel electrophoresis. as you can see here. Now with blast sequencing and blast analysis, as well as using a norovirus typing tool, it was determined that strain RD12 was in fact genogroup 2, genotype P16. And when we created a phylogram, we confirmed that RD12 was in fact genogroup 2, genotype P16, and clustered with other genogroup 2, genotype 16 norovirus strains. And now I'll move on to case 2, which was that of a family that had returned from vacation in which the mother became sick during the flight. And after that, when the mother recovered, the father and the son became ill with vomiting and diarrhea. And we collected fecal samples and labeled them RD15 and RD16 from the father and from the son, respectively. So similarly, we used those same universal primer pairs to detect norovirus. And in this gel electrophoresis, we saw amplicon size of about 319. Now, lane 2 and lane 4 are representing us testing the samples for rotavirus, in which they were negative for. So this slide is kind of compacted, but we also used the additional genotyping primers, and then we sent for sequencing and blast analysis, and it was shown that both RD15 and RD16 samples were that of genogroup 2, genotype P16 noroviruses. So to draw conclusions from these two cases, to our knowledge, this is the first report on the detection and molecular characterization of norovirus in humans from the Lesser Antilles. The norovirus strains from St. Kitts belong to genogroup 2, which is a common human norovirus genogroup. And previously, several clinical cases of norovirus-like diarrhea in humans, where they present with vomiting and diarrhea, have been reported in the RUS community as well as in the JNF hospital, but they remain undiagnosed because of the lack of laboratory tests. So considering the arrival of cruise ships, increased intake of seafood, and close proximity of humans, I suppose once the world opens back up, large-scale studies on the detection and molecular characterization of noroviruses in the islands of the Lesser Antilles is really important because it has implications on public health. Now, this last case I will present to you in this chapter will actually be about suppovirus. So I would like to talk about it here. This suppovirus causes acute gastroenteritis in humans and animals. And although outbreaks of gastroenteritis associated with suppovirus are less frequent than those with noroviruses, they do happen and they can be reported in all ages. And the transmission of suppovirus is through the fecal-oral route, and there is evidence of zoonotic potential of suppoviruses. They also belong to the family Policiviridae. They are positive-sense single-strand RNA viruses. They have been classified into at least 19 genogroups and 51 genotypes. Genogroups 1, 2, 4, and 5 have been shown to infect humans. So, although suppoviruses have been reported in humans with diarrhea worldwide, similarly to norovirus, there are no reports on the detection and molecular characterization of suppoviruses from the Lesser Antilles based on PubMed search. So that was the objective of this part of the study. So case 3 was that of a boy that was 3 years old that presented with vomiting repeatedly, a slight fever, had diarrhea, and we collected a direct fecal sample and we labeled that RD13. Again, same process. We used the universal primer pairs P289 and P290 to see if norovirus could be detected. Now, with the gel electrophoresis, we saw a similar amplicon pattern as the ones before. So honestly, in the lab, we were thinking, oh, another norovirus sample. However, let this teach you a lesson that with anything, you need to sequence and blast analyze, because this actually, in fact, was a supplevirus strain. And blast analysis showed that this was a genogroup 2 strain. And to further genotype this strain, instead of the MON primers, we had to use a different set of published primers that are used to amplify a short region of the ORF2 that encodes the capsid protein. And the amplicon size is about 412 base pairs, which is what we saw. We confirmed with blast analysis, and it was decided that RD13 was genogroup 2, genotype 3 supplevirus strain. And with a phylogenetic analysis, the confirmation of the genogroup and genotype can be seen here, clustering with other genogroup 2, genotype 3. This will lead me to my conclusions for this case. To our knowledge, this is the first report on the detection and molecular characterization of suppleviruses in humans from the Lesser Antilles. And the supplevirus strain from St. Kitts, again, belonged to genogroup 2, genotype 3, which is a common human supplevirus genogroup and genotype. The partial ORF1 as well as the partial ORF2 nucleotide sequences exhibited maximum homology to genogroup 2, genotype 3 human supplevirus strains, suggesting that strain RD13 may not be an intergenogroup or intergenotype recombinant, but we would have needed to do whole genome analysis in order to confirm this. Considering the lack of information on human suppleviruses from this part of the world and its great implications on public health, further stories are warranted in the Caribbean region. And and these are my references for this chapter. And I would just like to, so that was the end of my presentation. And as far as my master's project goes, I just wanted to show you and share with you some of the publications from my master project, two in which I already mentioned, being the titles of chapter 1 and chapter 2. But the PBV work was published in Viruses, and then the PCV2 work was published in Transboundary and Emerging Diseases, both within this year. So, and then we also had another journal article on PBVs in pigs, and I was a equal contributing author for that one, and that was published in Infection Genetics Evolution. And I was also an author on a chapter in Emerging and Transboundary Animal Viruses about tortanovirus. And these are some of my presentations during my master's project. I was very blessed and honored to be able to go to Barcelona, Spain in February before the pandemic hit. And I presented a poster on the PBV work at the Viruses 2020 conference, which was actually the same journal that we published the work in. So it was quite ironic and exciting. And then last year, I had an oral presentation at the St. Kitts Marriott for the ACAC PMP Congress, in which I presented chapter 3 of my project, which was the norovirus and supplevirus work. And then also before the pandemic hit, Dr. Ghosh went to New Delhi, India, and presented a poster on the PCV2 work in behalf of myself, which was really nice. And now I would like to acknowledge and thank several people, in which I want to go through this list and mention them individually, as each person and organization greatly helped our project. So first and foremost, I would like to thank Dr. Ghosh. Oh, okay, I'm being told to cut it out. Okay, well, I think I lost track of time a little bit. So anyways, all these people and organizations were very important in this project. So I just wanted to say thank you so much for everybody that made this project possible.